Hi, everyone. My name is Ishita Varman, and I'm a field application scientist with GenScript. Today, it is my pleasure to introduce our third speaker of the day, Dr. Branden Moriarty. Dr. Moriarty is currently a tenured associate professor in the Department of Pediatrics, Division of Hematology Oncology at the University of Minnesota. He holds a bachelor's degree in biology, chemistry, and biomolecular sciences from St. Olaf College, and a PhD in molecular cellular developmental biology and genetics from University of Minnesota. He did his postdoctoral work on cancer gene discovery. He then joined the Department of Pediatrics as faculty in 2014. Over the last decade, he has worked on developing a productive translation genome engineering program, which is focused on the development and translation of engineered immune cells for cancer and gene therapy. Please join me in giving him a warm welcome. The topic of his presentation is cytokine armored CAR-NK cell therapy for advanced ovarian cancer. Take it away, Dr. Moriarty.
Hi, I'm Branden Moriarty. First, I would like to thank the organizers for inviting me to present my work today. And today I'll be talking about cytokine armored CAR-NK therapy for advanced ovarian cancer. So we're focusing on the translation of a highly engineered CAR-NK cell. Our project goal is to develop, translate, and carry out a phase 1 clinical trial using highly engineered allogeneic CAR-NK cell therapy for the treatment of advanced ovarian cancer. And so you might be wondering why CAR NK cells, as there are many alternative immunotherapies available. We believe strongly that there are properties associated with NK cells that are advantageous for immunotherapy. For instance, they have an inherent cancer cell killing ability, so they can identify cancer cells with low MHC expression. They can also carry out ADCC against cells that are bound by antibodies, and they can also be equipped with CARs to further enhance their targeting and activation abilities. Moreover, in recent years, the engineering approaches for NK cells have been developed by our group and many others. Moreover, feeder-based expansion methods have been available for NK cells for some time, allowing greater than 10,000-fold expansion in 21 days. Moreover, they're an off-the-shelf or allogeneic therapy such that we can make large volumes of therapeutic cells and use them in many different patients. Moreover, graft-versus-host disease, cytokine release syndrome, and neurotoxicity are exceedingly low with NK cell therapies, as has been seen in the clinic, and this is compared directly with T-cell-based immunotherapies that do have instances of these adverse events. Also, epithelial ovarian cancer cells are susceptible to inherent NK cell killing, and clinically, the percentage function and IL-15 responsiveness of ascites-derived NK cells correlates with improved prognosis-free or overall survival at advanced stage epithelial ovarian cancer patients. And so as there's been a lot of interest in the use of NK cells for cancer immunotherapy, many investigators have looked at different sources. One of the first sources, of course, is just from peripheral blood of humans or healthy donors. Others have worked with iPS or ES-derived NK cells, and some groups are even using transformed human NK cell lines like NK92. In our group, we really focus on the peripheral blood NK cells as we believe they have a true NK cell phenotype. They're also readily available from healthy donors that you can screen. They're non-transformed in nature, and there's no cost and complexity of deriving them from iPS or ES cells. And as I mentioned previously, they can be activated and expanded using derived feeder cells, and they've been used in numerous clinical trials with excellent safety profiles. And so for our engineered CAR-NK product, we've decided to target the cancer antigen mesothelin, as we believe this is a good target for ovarian cancer therapeutics. So it's a well-established tumor antigen expressed in greater than 85% of ovarian cancers. It has relatively low expression in normal tissue, and it's also been targeted in a number of clinical trials using both antibody-drug conjugates and CAR T cells with relatively low toxicity. Additionally, we'll be cytokine armoring our engineered CAR-NK cells to enhance their function and persistence. There was some really nice clinical work from Katie Rezvani's lab showing that using a CD19 targeted CAR-NK product with soluble IL-15 expression could actually prolong the in vivo persistence of NK cells. So historically, we've seen less than 3 weeks of persistence in clinical trials and animal models using NK cells. But in this particular clinical trial where they performed cytokine armoring with soluble IL-15, they were able to detect the gDNA of engineered cells for up to 1 year in these patients. Moreover, similar findings have been developed or published in preclinical animal models, also demonstrating that using IL-15 cytokine armoring can enhance the persistence and function of NK cells. And then the last moiety that we're utilizing in our engineering approach is a synthetic cell surface receptor called RQR8, which acts as both a suicide gene and selection marker. So the suicide gene component is actually an epitope from CD20 that can be targeted with rituximab as a suicide switch. The other moiety in the cell surface receptor is CD34, and that can be used for the GMP compliant Clinimax CD34 immunomagnetic enrichment if desired. So as I mentioned, we're focusing on translating this highly engineered CAR NK cell product with these three different moieties that I just covered. And so here's our design construct that we plan to utilize to engineer NK cells. So we're including a strong MND promoter to drive the transgene expression. MND is a synthetic promoter that works very well in lymphohematopoietic cells. And then, of course, we have a CAR targeting mesothelin to enhance the activation of NK cells upon interaction with mesothelin-expressing cancer cells. And then the RQR8 synthetic receptor and suicide switch. In addition, the cytokine armoring with soluble IL-15 expression. And so historically, many groups have used retrovirus or lentiviral approaches to engineered immune cells for cancer immunotherapy, but we've actually focused on transposon, non-viral based genome engineering. And for those of you that are unfamiliar, DNA transposons are a non-viral, simple approach to stably integrate genetic cargo. Essentially, it consists of a DNA transposon and then a transposase enzyme that is delivered in trans as a synthetic mRNA. These are electroporated into the cells of interest where the transposase can identify the transposon through ITRs or inverted terminal repeats. And then it is randomly integrated into the genome and stably expresses the cargo encoded in that transposon for the life of that cell. Indeed, transposons have been used in a number of clinical trials to date, largely focused on human CAR T cells. So as you can see on the left there, there's a review of the many clinical trials in academia using this. On the right are two companies that are actually deploying transposons for engineering CAR T cell, and that's Poseida Therapeutics and Luminary Therapeutics. And I was actually able to participate in a IND application, which was successful with Luminary Therapeutics, where they took from concept to clinic a BAFF ligand-based CAR T cell product engineered with a transposon in less than 18 months. And so the transposon system that we utilize is actually called TC Buster. So this is a naturally occurring DNA transposon system, but we actually focused on evolving it into a hyperactive system for engineering immune effector cells. One of the main reasons that we were interested in this is some of the other available systems like Sleeping Beauty and Piggyback DNA transposons are IP restricted, very much so inhibiting commercialization. And so we performed structure-guided and in vitro evolution of the TC Buster transposase to generate a hyperactive variant called TCBM. And then we further deployed this and optimized the generation of both CAR NK and CAR T cells from peripheral blood immune effector cells, where we can easily achieve greater than 50% in engineering without any selection. We also found from this work that optimal transposition or transposon integration really requires a minimal backbone plasmid. And for these purposes, we've actually deployed the GenCircle technology that's recently been developed by GenScript. And so, what are some of the pros and cons of using transposons compared to the historic viral delivery approaches? So, both of these can mediate stable gene transfer. They've both been used clinically. Cargo sequences, some of them can be limited for viral delivery, like splice acceptor sequences, polyadenylations, where there are absolutely no cargo sequence limitations for DNA transposons. Another huge attribute for the transposons is that they can integrate very large cargo, easily 12 kilobases in size, whereas there's variable size capacity with lentiviral, retroviral, and AAV. Insertional mutagenesis is a concern with any randomly integrating vector. Viral approaches really have preferences for active genes, whereas transposons, it's really dependent on the transposon system. And with all three of the DNA transposons that I've discussed previously today, they all have a safer integration profile than lentivirus or retrovirus. Another huge boon to transposons is the cost. So it's about 2 to 5 times less expensive to acquire the GMP reagents for engineering immune effector cells with transposons as opposed to virus. Historically, there have been concerns with DNA toxicity, so you are delivering DNA plasmids to immune effector cells that have evolved mechanisms to sense DNA in the cytosol to avoid bacterial or DNA virus infection. But actually, through some methodologies that we and others have developed, we've overcome a lot of this DNA toxicity. Lastly, using the hyperactive transposases, we can get very high efficiency, comparable, if not better, than viral approaches. And as I mentioned, they are significantly cheaper. So you can obtain these GMP transposons in less than 4 to 5 months for around $400,000 from many vendors, including GenScript. And this is enough plasmid to actually create over 1,000 doses of immune effector cells. Moreover, for the TC Buster system, the GMP transposase mRNA is readily available off the shelf for sale from Bio-Techne. And so, getting back to our specific project, then, our first step was a selection of an optimal mesothelin CAR to generate our cytokine armored CAR NK cells. And so, for this, we looked to the literature and we identified 3 different binder moieties that we could utilize in the CAR architecture. The first was a SS1 scFv derived from the NIH many years ago. This is a murine derived binder. And then we identified an M912 scFv, a humanized scFv binder. And then we also identified a nanobody-based binder from Camelid called 121 nanobody. And so all of these contained a transmembrane domain, 41BB co-stim, and CD3 zeta stimulation domains. We then derived transposon vectors that encoded all of our elements that we covered earlier, so the CAR, RQR8, and soluble IL-15, all encased within a single transposon vector. We then generated primary human peripheral blood NK cells overexpressing these constructs. You can see here by these flow plots that we get very good stable integration without selection, achieving upwards of 98% integration. We then took these engineered NK cells and performed in vitro co-culture assays with mesothelin high and mesothelin low expressing cell lines of ovarian cancer. And in this case, we found that the SS1 and the 121 binder induced significantly upregulation of degranulation, interferon gamma, and TNF alpha, both in the mesothelin high and in the mesothelin low line. And so that was good validation in vitro that at least a subset of these different CARs was functional for activation. Next, we then moved all 3 of these candidate CAR constructs into a pilot in vivo study. So here on the bottom is just a diagram of how the study works. So we took immunodeficient NSG mice, injected A1847 ovarian cancer into their IP space orthotopically, allowed those to grow for 14 days, randomized on day 7, and then we treated them with a single infusion of 5 million NK cells. That would be the cytokine armored CAR NK cells, or as a control, just NK cells alone. We did a bleed on week 6 to look at peripheral blood persistence of our NK cells and also at endpoint. And so here's the results of that study that lasted about 15 weeks. So you can see the bioluminescence images over on the left, and that's quantified on the right on the top, where you can see that tumor control was achieved with all of the CAR groups, albeit the SS1 CAR performed significantly better than the M912 or the 121 CAR. You can see having survival well out over 100 days. We were also then interested at the 6-week blood draw to look at persistence of NK cells. With the NK cells alone in the M912 CAR-NK, we actually didn't see a persistence of NK cells. And with the M912 construct, it could be that it's not expressing high levels of IL-15 or other issues with that construct. But actually, with the 121 and SS1 construct, we saw very high rates of peripheral blood NK cells at 6 weeks. And as I noted earlier, other studies published in the literature that do not provide gamma cytokine chain, either IP to the animals or encoded within the NK cells, they do not see NK cells persisting longer than 3 weeks in animal models. Whereas here, now 6 weeks out, our cytokine armored NK cells are still robustly present in the peripheral blood. And then, as I mentioned, the most interesting group was the SS1 group, which appeared to face value actually eradicate all of the ovarian tumor within the IP space. And if you look at the middle group there at week 8 and week 15, you can see on the right, there's completely an absence of bioluminescence imaging. And then on the right, we see obscure bioluminescence signal on the bottom right of the abdomen. And when these animals are necropsied, we see that it's actually an artificial tumor that's caught in the IP space. We hypothesize that this is from using the same needle to inject the animals when we're orthotopically implanting the cancer cell line. And so we've now switched to using new needles between injections, and we've resolved this artifact. And so based on these in vitro and in vivo studies, we plan to proceed clinically with the SS1 CAR for our clinical trial. And so, in order to enable our ability to carry out this clinical trial, we've also developed a GMP-compliant transposon engineering process for peripheral blood NK cells, and that's depicted here on the top of the screen. So essentially, we isolate peripheral blood NK cells through Clinimax isolation, and then we do a co-culture with irradiated feeder cells that were developed by Dean Lee that essentially express membrane-bound IL-21 and 4-1BB ligand. We activate them for 5 days in the presence of IL-2, and then we perform electroporation, delivering the mRNA encoding the transposase and the GenCircle plasmid encoding our transposon. These are then immediately put into a second expansion, again with irradiated feeders, out to day 12, where we can analyze them for protein expression, viability, and fold expansion. And so here's just some results that we generated in our optimization. We're on day 12. We had about 55% of the cells expressing our CAR of interest. Then at this point, we put them into a final third expansion to generate enough numbers to carry out the clinical trial. And so we've done the calculations, and based on this, we can use a single leukapheresis unit, and we can produce enough CAR NK cells for our dose escalation study to treat 29 patients. So in the second leg of this manufacturing, we have the option to do the Clinimax enrichment using that RQR8 modality that I mentioned, so CD34 positive selection. And then they can be placed into a third expansion with irradiated feeders. In this case, we use one of the G-Rex flasks, or the rapid expansion gas permeable flasks. These are then grown out to day 19, analyzed and cryopreserved for clinical use. And again, in one of our recent studies, we achieved greater than 95% of the NK cells having expression of the CAR of interest. And notably, we achieve over 2,000-fold expansion in this 19-day process. And so with that, just some conclusions and future directions. So we've determined that DNA transposons are a safe, affordable, rapid, and efficient mode of immune cell engineering. Further, we've developed GMP-compliant manufacturing processes to generate CAR NK cells. Our mesothelin CAR NK cells are functional in vitro against mesothelin high and low ovarian cancer cell lines, and the soluble IL-15 expressing CAR NK cells, or cytokine armored, can persist for long-term in vivo. And again, cytokine armored SS1 CAR-NK cells can visually cure ovarian cancer in some mice and significantly extend life in others. And so in our future directions, what we're working on right now is a comprehensive IND enabling study of these armored CAR-NK cells. So we had submitted a pre-IND last year, and so we did receive feedback from the FDA on what they expected in our IND package, and so we're generating those data packets currently. And then we also are obtaining our GMP GenCircle encoding our TC Buster transposon. So we ordered that earlier this year, and it's slated to arrive at our GMP facility in September of 2024. Then we'll be working on technology transfer of our GMP compliant CAR-NK manufacturing, and that will take place in Q3 of 2024. We will then work with our GMP facility to produce engineering runs, scaled runs, QC testing for purity, sterility, and stability by Q3 or 4 of 2024. We will then take all of our preclinical data, IND enabling studies, and GMP manufacturing run data, compile those into an IND application, and submit that to the FDA to be able to carry out a phase 1 trial to treat advanced ovarian cancer patients with cytokine armored CAR-NK cells. And we hope to submit the IND by the end of 2024, with initiation of the trial in early 2025 and completion by the end of 2026. And with that, I would just like to acknowledge everyone that's been part of this work. So all the researchers in my lab, particularly our postdoc, Jay-Woo Chang, and my graduate student, Josh Kruger. This is all part of a NIH SPORE grant, and my collaborators there are Dr. Bo Weber, Melissa Geller, Tim Starr, Jeff Miller, and Martine Felices. And thanks to our collaborators at Mayo Clinic, Scott Kaufman and Jamie Bacum-Gomez, who are the main PIs of the SPORE grant, and to our GMP facility at the University of Minnesota. And with that, I will stop and take any questions.
Thank you so much, Dr. Moriarty. Really, I think it was enlightening to see you bring CAR-NK to the forefront, since CAR-Ts have stolen a lot of the limelight. Also, the use of our products, GenCircle, particularly for your transposon engineering of the NK cells, that was quite a learning lesson, I'm sure, for the audience as well, not only for me. So thank you once again. Having said that, we do have quite a few questions. I'll start with the first one. Given the promising results with SS1 CAR-NK cells in your in vivo studies, what additional modifications or strategies are being considered to further enhance the efficacy and safety of the cytokine armored CAR-NK cells for treating solid tumors, particularly in preparation for the phase 1 clinical trial?
Yeah, it's a good question. And for the phase 1 clinical trial, we're so far along that we're not incorporating any additional engineering steps. And of course, we've already ordered our GMP GenCircle, so that is kind of set in stone. In other projects in the lab, we are combining transposon engineering with multiplex base editing using adenosine base editors, and we have a preprint up for that. Where we can edit up to 6 genes simultaneously while also introducing a transposon construct. But in addition to that, we are looking at modifications to culture conditions. That is something that we still have time to change for the phase 1 clinical trial for ovarian cancer. And so there are some media supplements that we're testing that look very promising for reprogramming the phenotype of NK cells that enhances their function and persistence, and that's something that we're looking at possibly incorporating for this phase 1 trial.
Thank you so much. The second question is, can there be any application of retrotransposons?
Yeah, I mean, that's another great question. There's a lot of interest in both retrotransposons and the recently described CRISPR transposon systems. There is a retrotransposon called Hellraiser that has been shown to mediate stable gene transfer in cells that is of interest. And then some of the products, actually, I believe, developed by Tessera Therapeutics, are based on retrotransposons in nature. And so there is some site specific integration being utilized there as well.
Thank you. The third question is, have you looked at the NK cell in different organs at week 6, injected with different CAR NKs?
Yeah, we haven't looked at a lot of different tissues. We've looked at the main tissues where we'd expect to see NK cells like the bone marrow, spleen, peripheral blood. But in this model, we have not looked at other tissues. In other studies in the lab using more highly engineered NK cells where we see some toxicity, we do see them in many other tissues, including the lungs and kidneys and other tissues when they're having lots of in vivo expansion. But in this model, we haven't dug too deeply into that yet.
Thank you. Fourth question, how do you see the future with NK CAR cell therapies? Do you think NK92 has a long term future in comparison to primary NK cells?
Yeah, I think they both could be utilized long term. Obviously, there could be safety concerns with using cell lines like NK92s, but I think both of them are applicable. And really, I don't think that either of them on their own will cure cancer. I think that they're a vehicle to induce secondary immune responses and that will come in and really clear up cancer. This has been seen even with CAR T cells, where it actually, in solid tumor models like osteosarcoma and rhabdomyosarcoma, it isn't the CAR T cells that induce durable responses, but they found that endogenous T cell responses through T cells when they're TCRs that are reactive to tumor antigens are really what come in and clean this up. And that's why we're really excited about using soluble IL-15 as our cytokine armoring, because it's known to activate other immune cells in addition to just the cytokines that NK cells naturally produce to induce secondary immune responses. But in the end, I think if it's a transformed cell line like NK92 or primary NK cells, I think you could have the same therapeutic benefit.
Thank you. The last question from the audience side is, what is the future direction?
Yeah, I think the future direction for the NK cells is, further enhancing their function and persistence in vivo. And so, like I mentioned, combining it with multiplex base editing to edit genes that are even cancer cell type specific. So, tailoring the engineering to an exact cancer type of interest, whether that be, knocking out PD-1 or TIGIT or other molecules that have cognate receptors on the cancer of interest, and then enhancing their in vivo persistence and their ability to initiate secondary immune responses, I think will be critical to the effective future of therapeutic NK cell therapies.
I think we do have time for a few more questions. A few additional questions came in as well. The next question is, I see you deliver the CAR NK via IP injection in your model, but won't these be delivered as IV in patients?
Yeah, interestingly, for our clinical trial, we'll be working with Dr. Melissa Geller, who's one of the clinicians here at the University of Minnesota. And in her previous trials using less modified NK cells for ovarian cancer, she actually delivers them via IP injection. So they actually put in an IP portal and we're able to deliver them directly to the cancer milieu in the IP space of the patients where the ovarian cancer exists. And so we will be delivering at IP in human beings as well.
Interesting. The next question is, have you tested your CAR NK cells in other animal models like PDXs?
We are ramping up to do some testing of this particular product in PDXs, but we have parallel programs targeting other cancer antigens like CD70, where we've done a number of PDX models in renal cell carcinoma and then xenografted AML models as well. And we'll be publishing that later this year.
Thank you. And I believe this will be the last question. Have you observed any toxicity, particularly with the IL-15 armoring?
Yeah, like there was a publication from Bonifant describing a cytokine armored NK cell where they did see some toxicity. With this particular construct, we don't see toxicity from the cytokine production, but in some of our other NK models, including um, some leukemia models where we get a really large expansion of NK cells in vivo and the IL-15 levels become much higher. There we do see toxicity in the immunodeficient mouse models. Although it's hard to know if that will really occur clinically, because of course in these NSG models, they don't have other immune effector cells that would really sop up soluble IL-15 or, reduce the reservoir of IL-15. So we are interested in looking at humanized mouse models to see if the toxicity is really going to be a problem in like a full immune system background. But yeah, we have seen it in some models.
Thank you so much. With that, I believe we are at the end of our talk. Do like to thank you so much for not only doing this work, bringing CAR NKs to the forefront, but also delivering such a fantastic presentation.
Thanks.
With that, we are transitioning into a break, and we will see you for our next session after the break around 1:50 p.m. Eastern Time. So please do log in a few minutes earlier. Thank you, guys.